So this session is related to experimental technique exercise 3, which is a weekly homework as well. In the first question, which is from paper 4, the structure question, a list of techniques used to separate mixture is given below. So you can have a chromatography, crystallization, diffusion, evaporation, filtration, fractional distillation, and simple distillation. From the list, choose the most suitable technique to separate the following. If we need water from seawater, basically seawater is a salt solution. So if we need water from seawater, then which technique we will use? We'll use simple distillation. We want to separate helium from a mixture of helium and methane. So if you want to separate two or more gases, because different gases have different rate of diffusion, so here we can write diffusion, or we can also use fractional distillation. So both are acceptable, as in the option we have both diffusion and fractional distillation. So any one of them we can write. If you want to separate two or more miscible liquids or two or more gases which are mixed together, we can use diffusion specifically for gases, and fraction distillation, if they are miscible, they mix with each other. So first we convert the gas into liquid and then separate them on the basis of the boiling point. Ethanol, from mixture of ethanol and propanol. So two alcohols are there, they're completely mixed together as they're both liquids. So whenever two or more miscible liquids are there, so what we can use? We can use fractional distillation. Iron fillings, from mixture of iron filling and water. So iron filling means small pieces of iron. So iron is insoluble. As you add iron metal to water, iron does not dissolve in water. So if you have insoluble solid, so which technique you can use? You can use filtration. And amino acids are colorless liquids. So for amino acids, colors, dyes, food colors, what we use? We use chromatography. Is it clear? Question 1. Anyone having a doubt? So water from seawater, we use simple distillation. Mixture of gases, we use diffusion or fraction distillation. Alcohols are mixed together, we use fraction distillation. Iron fillings are insoluble in water, so we can filter them out. And a mixture of amino acids, so we can use chromatography. Then in question 2, they're asking what is meant by a term fractional distillation. So basically, you have to write the definition of a fraction distillation. So fraction distillation is simply separating the substances on the basis of boiling point. Separating two or more miscible substances on the basis of boiling point. This technique is known as fractional distillation. In question 3, a list of techniques used to separate the mixture is given below. The filtration, diffusion, fraction distillation, simple distillation, crystallization, and chromatography. From the list, choose the most suitable technique. A technique may be used once, more than once, or not at all. So it's not necessary that all of them will be the answer. So you can use them more than once, or not at all. So butane, from a mixture of propane and butane. So you have two, or basically both are the gases. So when we have gases, the technique which we can use diffusion, or we can use fractional distillation. So when two or more miscible substances are there, we use fractional distillation. Or how diffusion can be used, because when two gases have different masses, as there are different gases, so they will diffuse at a different rate. So diffusion is not 100% accurate, but it can be used, as the two gases will have different masses, so they will diffuse at a different rate. So what we can do, we can put a barrier, like we have a container. The container is a porous container. Porous means like it is having small holes, and the gas is inside, the mixture of a gas. So which gas will come out first? The gas which is lighter will come out first. So, and the gas which is heavier will remain in the container for a longer period of a time. So this technique is called diffusion, but it's not 100% accurate. Some of the heavy gas will also come out, but it can be used. Oxygen from liquid air. So if we are separating oxygen from liquid air, again the mixture of gases, and here we have a liquid air, so that's why we cannot mention diffusion, because diffusion is only valid for gases. But fractional distillation, when a gas is converted into liquid and then we separate on the boiling basis of boiling point, so we call that as a fractional distillation. Water from aqueous magnesium sulfate. So we need water from salt solution. When we need water from the salt solution, so the technique which we can use, that is called simple distillation. Potassium chloride from aqueous potassium chloride, like you have a salt solution, you have a salt dissolved, because aqueous potassium chloride means a salt solution. So you have a salt solution and you want salt from this, so what you can do, you can use evaporation or crystallization. So only crystallization is mentioned, evaporation is not there as an option, so we can mention here crystallization. Fractional distillation, Abdullah, is used normally for two or more miscible liquids. But here you have water and the salt. Salt is solid, which is dissolved, and you need water out from that. That's why it is simple distillation. So if you want a liquid from a solution, a salt solution, then it is simple distillation. A silver chloride from a mixture of silver chloride and water. So as we discussed, most chlorides are soluble except silver, mercury and lead. So this is insoluble, so this will be solid. So right now we don't have a salt solution. Basically what we have a container which is filled with water, and we have a solid silver chloride. So we have this solid silver chloride excuse me, and we want to separate it from the water. So what we can use, we can use filtration. And glucose from a mixture of glucose and maltose. So for identifying if it is a dissolved one, you can mention chromatography, or you can mention if it's a solution. Normally the correct answer is chromatography, but other one, chromatography in that sense that you have a sample or a mixture of a dissolved sample of glucose and maltose. So you, in that case, you can use chromatography. Is it clear? Question 3. Anyone having a doubt? Any question or a doubt in 3? Yes, Abdullah?
Sir, in D,
Hmm.
can't we use the evaporation process?
Yes, we can use evaporation, but in the list they did not mention evaporation, because in the question they mentioned from the list choose the most suitable. So in the list evaporation is not there. Otherwise, yes, you can mention evaporation.
Okay, sir. Thank you.
Then moving on to question 4, the following techniques are used to separate the mixture. Simple distillation, A is simple distillation, B is fraction distillation, C is chromatography, D is filtration, E is evaporation, and F is diffusion. From the list, most suitable technique, methane from mixture of gases, methane and ethane. So when you have gases, so you can write diffusion. So diffusion is denoted by D. You don't have to write the name. You just have to select a letter for this. So diffusion is D, or it can be fraction distillation. So it can be B. So both are possible, but you don't have to write both of them. Any one of them. Diffusion is F. Sorry, I diffusion is F, and fraction distillation is B. So it can be F or it can be B. Water from aqueous magnesium sulfate. So you need water from a salt solution. Aqueous means salt solution. So if we need water, we can use simple distillation, that is denoted by letter A. A mixture of amino acids. So amino acids are colorless liquid. So we use chromatography. Iron filling, which are insoluble in water. So we can use filtration. So D. Zinc sulfate crystals from aqueous zinc sulfate. So we need salt from solution. So when we need salt from the solution, so we can use crystallization or evaporation. But here there is no option of crystallization. So evaporation is denoted by option E. Hexane from a mixture of liquid, hexane and octane, two or more miscible liquids are there. So what we can use fractional distillation. Is it clear? Question 4. Abdullah, you have any question? Abdullah Safras?
Yes, sir. Can you explain number E?
Hmm. Zinc sulfate from aqueous zinc sulfate. What is the meaning? It means you have a salt solution, you have a salt zinc sulfate dissolved in water. So you have a salt solution and I want salt from this. So if I have a dissolved salt in water and I only need salt from this, so what I can do, I can either evaporate, means I can supply heat energy until all of the water will escape, and what we are left behind when all of the water escape, so we are left behind with the salt. That's called evaporation. Is it clear?
Yes, sir.
In question 1 related to alternative to practical ATP, a fake banknote can be investigated by dissolving the ink of the paper. So there is a fake banknote, and we can investigate its ink by dissolving in water. You are provided with 4 different inks. So there are 4 different inks from 4 different criminals. So 4 different criminals, you have a sample of 4 different inks. Describe an experiment to show which one of these inks is the same as the ink from the banknote. And you can use a label diagram to help your answer. So simply what we will do, I will write the answer, but first explanation. So what we'll do, we'll use a chromatography. And how we carry out this chromatography, so this is a chromatogram which we will take. Then we will draw the origin with pencil. Here we will place ink from the banknote. I'm writing BN. So ink from the banknote, that is BN. Then we will take, and we have inks from 4 different criminals. So example, from criminal 1 we have C1, criminal 2 C2, criminal 3 we have ink 3, and criminal 4 we'll have C4. Then we will use a suitable solvent which can dissolve these inks. So as the chromatogram will absorb the solvent will rise on the chromatogram. How we can identify that which uh, criminal is having the banknote ink? So the one which matches with the this is a spot of ink from criminal 1, criminal 2, criminal 3 and criminal 4. So which spot matches with the banknote ink? So this is a spot of a banknote and this is a spot from criminal 3. So it means it matches with criminal 3. So it means this banknote ink belongs to criminal 3. Yeah, it is very important you draw the diagram so that it will clear the, if you miss anything in the question, it will clear, complete the answer. That's a reason why you should always draw. So basically, but you don't have to say which criminal is having, you just have to outline the experiment, that how it can be done. So what we will do, so first thing we will use chromatography, and then we will draw the origin with pencil. Then we will place ink from banknote, and inks from different criminals. And then what we will do, we will use a suitable solvent here, like you can mention water. Then we will compare the height of ink from banknote with the criminal samples. The one which rise with same rise to same height belongs to the criminal. So we can check the height, the position, compare, and we can identify that which one belongs to that criminal. Is it clear? The experiment. So we have, like, there's a fake banknote, not original banknote, and there are sample of inks from four different criminals. So what we did, we take a ink from the fake banknote, and we took the ink from four different criminals, and we carry out a chromatography, and we check the height through which the ink from the banknote and ink from the different criminal rise. The one which matches or rises to the same height, it means that belongs to. So this fake ink or fake banknote belongs to criminal 3 in this example. But you don't have to mention a specific criminal. You just have to outline the experiment. Is it clear? To everyone? So the next question, you can mention RF value. You can mention, like, compare So you can mention the RF value as well, like measure the RF value from the banknote and compare with the RF value of the criminals. You can mention that as well. So both ways it is right. Another question, in question 2, leaves are leaves from tree containing a mixture of colored pigments, like leaves having different colors and which are not soluble in water, so we cannot dissolve them in water. The student was given these two instructions to investigate the pigments in the leaf. So first instruction is crush some leaves to extract the color pigment. So first thing, he want to crush and then extract the color and use the liquid extract to find the number of colored pigments. So what you have to write the answer for first one. What would the student need in order to effectively carry out instruction 1? So how he can crush them and extract the color. So we can use mortar and pestle to crush the leaves, and then we will boil with alcohol because it is not water soluble. So we can boil in alcohol to extract the color, and then we will filter to remove insoluble particles. Then how we can identify how many colored pigments are there? So for that purpose we can use chromatography. Then draw origin with pencil, then use suitable solvent, not water. You cannot mention water, as I mentioned it is insoluble in water. Then wait for solvent to rise, then identify number of spots on chromatogram to know number of colored pigments in green leaves. Is it clear? So first instruction, we use mortar and pestle to crush it, and then we boil in alcohol. Why we boil in alcohol, not in water? Because they mentioned it is not soluble in water. So we boil in alcohol, then we filter. So that was a colored extract which we obtained. And then that colored extract we place on the chromatogram. We draw the origin with pencil and we identify number of spots. Like example, if two spots are there, it means it contains two pigments, two colors, colored pigments. If it is having three spots, then it is three color pigments. If it is one spot, and it means it is only one color. Is it clear? Question 2. And moving on to question 3. So they mention there is E110 is a sunset yellow. So it's basically it's a food color, E110. You can find different types of food color, and these are actually the codes for the food colors. You can find different food colors having different numbering. So E110 is basically a sunset yellow. outline a method you could use to show the presence of E110 in a food coloring. So there's a food coloring and you want to show that it contains E110. A space has been left if you want to draw a diagram to help your answer help you answer the question. So you can draw a diagram if you want to help you answer the question. So what we will do, we can do in two manners. We can take a chromatogram, draw the origin with pencil, and then we will place a food color. So food color I'm writing like FC, and then we will take E110, the color, the actual E110. We want to outline a method to show the presence of E110 in a food coloring. So food coloring contain E110. So how we can work out? So we'll use a suitable solvent. So when you use a suitable solvent, the chromatogram will soak up the solvent and the solvent will rise on the chromatogram. So if the spot from the food coloring and spot from E110, they rises to the same height, or they have the, or we can measure the RF value for both. If they have the same RF value, it means the food coloring contain E110. So the answer for this, like writing in words, so we'll use chromatography. Then we'll draw the origin with pencil. Then we'll place food color and E110. We'll use a suitable solvent. Uh, when you mention in exam, don't mention use suitable solvent. You will mention the name of the solvent. Like use suitable solvent. Example here we are using water. Then wait for solvent to rise. Then measure the RF value or compare the height from food color and spot for E110. If they rise to same height, or it having a same RF value, it means that the food color contain E110. Is it clear? Discussion 3. Any doubt or a question? So this was exercise 3, which was a weekly homework related to experimental techniques.